class. In this video, I am going to teach you about structure and function of Zona pellucida. In addition to that, I will elaborate you acrosomal reaction in mammals. If you are interested, then please continue watching till the end of video. At the end, I will solve some previous year CSIR net questions. So let's get started. How the acrosomal reaction in mammals gets initiated? For that, first the sperm have to bind with the Zona pellucida. After binding of the sperm to Zona pellucida, it leads to acrosomal reaction. This diagram represents the steps of fertilization. In the first step, sperm activated by female reproductive tract, that is translocation and capacitation of sperm. I have a video on that and given the link in the description box. Please have a look. After the sperm activated by female reproductive tract, sperm bind to the Zona pellucida. Then acrosomal reaction takes place. Now comes to Zona pellucida. What is Zona pellucida? Zona pellucida, this is a glycoprotein matrix which is synthesized and secreted by oocyte. Zona pellucida, it is the layer that is present outside to the plasma membrane of egg. Okay, it is present outside the plasma membrane of egg. What are the function of Zona pellucida? Zona pellucida has two functions. The first function is to bind with the sperm and the second function is the initiation of acrosomal reaction. Now comes to the structure of Zona pellucida. Zona pellucida made up of three major glycoproteins, that is the Zona pellucida 1, Zona pellucida 2 and Zona pellucida 3. Out of three Zona pellucida, the Zona pellucida 3, it binds to the sperm and initiate acrosomal reaction. Which Zona pellucida binds and initiate the acrosomal reaction? That is the Zona pellucida 3. Okay, here in the diagram, as you can see, that blue color, it is the ZP3. It binds to the ZP2. Okay, they are arranged in repeating units, and sometimes ZP3 and ZP2 repeating units, they are cross-linked by ZP1. And also you can see that on the surface of the ZP3, the red marks, that are the carbohydrate residues. That carbohydrate residue which is present on the surface of ZP3 is called as N-acetyl-glucosamine. Okay, what is that? That is the N-acetyl-glucosamine. And where it is present? It is present on the surface of ZP3 of Zona pellucida. The next is the binding of sperm to Zona pellucida. How sperm binds to the Zona pellucida? The sequential interaction between sperm protein and the component of the zona that leads to the binding of sperm to the Zona pellucida. So what will happen? In the first step, the sperm, it will weakly bind to the protein that are secreted by oviduct. Okay, the first step is the weak interaction of sperm to the protein secreted by the oviduct. Then the sperm surface protein SID1, which is presented in the yellow color, it will bind to the Zona complex. It will bind to the Zona complex. Then in the third step, the sperm galactoside transferase, that is the GALT, it cross-linked tightly and specifically to N-acetyl-glucosamine sugar residue present on ZP3. GALT, it will bind tightly to the N-acetyl-glucosamine residue on ZP3. So how the sperm bind to the Zona pellucida? The first binding is weak binding, that is the sperm, it binds very weakly to the protein secreted by oviduct. Then the SID1 protein of sperm, it will bind to the Zona complex. Then the GALT, it bind tightly and specifically to the N-acetyl-glucosamine on ZP3. So here in the diagram, this is the sperm with the SID1 protein, it is present on the surface, and GALT, it is present in the head of the sperm. So what will happen? The SID, it will bind to the Zona complex, and GALT, it will bind to the N-acetyl-glucosamine on ZP3. So what are the important points to remember? In addition to that SID1 and GALT, another set of protein that bind to the Zona are ADAM3 and ADAM2. This protein forms complex on the top of sperm head, and the sperm deficient in either one cannot bind to the Zona. Okay, So for binding to the zona, the sperm have seed 1, galled, Adam 3 and Adam 2. Similarly, on the egg, the Zona pellucida, similarly on Zona pellucida surface have N-acetyl glucosamine. Removal of N-acetyl glucosamine or the deglycosylation of N-acetyl glucosamine on ZP3 abolishes its ability to bind sperm. Acrosomal reaction. Acrosomal reaction in mammals occurs only after the sperm it binds to the ZP3 of Zona pellucida. Okay. The important point to be noted, how the acrosomal reaction in sea urchin and mammals they differ from each other. The difference is because of the thickness of extracellular envelope surrounding the egg. In sea urchin, the extracellular envelope is called as the vitelline membrane. This vitelline membrane is very thin. Vitelline membrane is very thin and porous, so that the sperm can easily reach to the egg. Okay, so sperm can easily reach to egg. That's why in sea urchin, acrosomal reaction takes place first. After acrosomal reaction, the sperm bind to the egg. But in case of mammal, the extracellular envelope, that is the Zona pellucida, it is very thick. It is very thick. That's why fast binding of sperm to Zona pellucida is required. After the binding of sperm to the Zona pellucida, then it initiates the acrosomal reaction. So what will happen? First, the galt, it is present on the sperm surface. Okay, it binds to the N-acetyl glucosamine on ZP3. When galt bind to the N-acetyl glucosamine of ZP3, that is the binding of sperm to the Zona pellucida, it will activate phospholipase C. Okay, it will activate phospholipase C. Then this phospholipase C, it will split phosphatidyl inositol 4,5-bisphosphate into 1,4,5 inositol triphosphate IP3 and diacyl glycerol. This IP3, which is formed by splitting of PIP2, it will release the calcium ion from endoplasmic reticulum. When the calcium ion it get released from the endoplasmic reticulum, that lead to the fusion of sperm membrane and adjacent acrosomal membrane. Okay. After the exocytosis of the acrosomal vesicle, the variety of enzyme will be released. One of those enzymes is N-acetyl glucosaminidase. N-acetyl glucosaminidase. And that lies the N-acetyl glucosamine residue on ZP3, so that sperm can travel towards the egg. Okay. So when the acrosomal reaction initiated, the anterior portion of sperm membrane that contain the region for binding to the ZP3 is shed from the sperm. Okay. But the sperm still have to travel through the Zona pellucida. The secondary binding of sperm to Zona is accomplished by protein in the inner acrosomal membrane that bind to the Zona pellucida 2. Okay. So before acrosomal reaction, the sperm membrane it bind to the ZP3 of Zona pellucida. But after acrosomal reaction, the inner membrane of acrosome it will bind to the Zona pellucida 2. Okay. Point to be noted, only acrosomal reacted sperm will bind to ZP2, but not the acrosome intact sperm. So here in the diagram, only one the sperm is able to travel through corona radiata and reach the Zona pellucida. In the Zona pellucida, the sperm it will bind to the N-acetyl glucosamine of ZP3. Okay. How it will be bind? Because the sperm has some proteins like a SID1, GALT, ADAM3 and ADAM2. Okay. Okay. So here is the diagram that describes acrosomal reaction. Only one sperm it will able to cross through the corona radiata and reach the Zona pellucida. After reaching the zona pellucida, the galt, that is the galactosyl transferase of sperm, it bind to the N-acetyl glucosamine of ZP3. After binding of galt to the ZP3, that leads to influx of the calcium ion, and calcium ion, it causes the fusion of sperm plasma membrane with adjacent acrosomal membrane, and that lead to the exocytosis of acrosome. It will release the enzyme. One of those enzymes is N-acetyl glucosaminidase that cleave the N-acetyl glucosamine present on the surface of ZP3 or after the acrosomal reaction, the sperm surface that bind with the ZP3, it will be shed from the sperm, and the inner acrosomal membrane, it will bind to the ZP2 of zona pellucida, because it have to pass through the zona pellucida layer to reach the egg. Okay, that's why the acrosomal reaction completed in mammal. Here is the previous year CSIR-UGC question. The question is, during fertilization in mammals, sperm egg interaction is mediated by zona pellucida membrane protein and their receptor present in sperm membrane. ZP3 has been identified to be the principal ZP protein whose post-translational modification is important for sperm egg interaction. In competitive inhibition assay, the sperm is saturated with either active ZP3 or its modified form. Before starting sperm egg interaction, which of the following experiment will not inhibit sperm egg interaction? In competitive inhibition, what happens? Both the substrate and the inhibitor, they compete with each other to bind to the active site. Here, the sperm, it is already saturated or bind with the ZP3. So, which of the following experiment it will compete with that ZP3, but that cannot inhibit the sperm egg interaction? The answer will be option B, deglycosylated the ZP3 protein and use it for saturation of sperm. We know that for the interaction or the binding of sperm to the zona pellucida, the zona pellucida membrane should have N-acetyl glucosamine on ZP3. If that N-acetyl glucosamine is removed or it is deglycosylated, then it cannot bind with the sperm. So, here if the ZP3 protein is the deglycosylated, then it cannot competitively or it cannot inhibit the sperm egg interaction. Okay. Thank you guys for watching my video up to the very end. If you found this video useful, then please don't forget to like, share and subscribe.